Eosinophilic gastritis and eosinophilic duodenitis are rare diseases with a chronic and often debilitating course, and are due to inflammatory cells like eosinophils and mast cells that cause inflammation in the stomach or small bowel. This leads to symptoms such as abdominal pain, nausea, vomiting, loss of appetite, and diarrhea. There are no approved treatments for these conditions. We conducted the first ever randomized controlled treatment trial using AK002, also called larentilimab, a medication that works through the Siglec-8 receptor by decreasing eosinophil numbers and deactivating mast cells, helping the GI tract to heal. Subjects who received active medication had nearly a 90% decrease in eosinophil levels in the GI tract compared to those who received placebo. Symptoms and treatment response were also significantly improved with the active treatment compared to placebo. Overall, the results showed that AK002 had strong benefit for treating these diseases, was well tolerated, and should continue to be developed.
We were nominated for this prize with a publication that was published in early June in Nature that described the convergent evolution of anti-SARS-CoV-2 antibodies in individuals who recovered from COVID-19. The way we studied this immune response, it all starts with a single blood draw. So we were able to enroll over 150 individuals here at Rockefeller University from April into early May, which was not an easy task because back then there was the peak of the pandemic in New York City and the healthcare systems were at capacity and was very difficult. And we then assembled this team of physician scientists, virologists, immunologists to all work together and to tackle this problem. And the method that we used initially is we take the blood from those recovered individuals and we're looking for antibody-producing immune cells. And the way we do this is we take a small fragment of the virus, we label it, and then we use this as bait to isolate those anti-SARS-CoV-2 specific B cells. And then we use a cell sorter, which is a quite impressive machine. It's able to isolate individual B cells into small, tiny Petri dishes. And once we have them isolated, we can look at the genetic information of these cells. And throughout the summer, we went through preclinical testing, and by the end of the year, we were able to start a phase 1 first-in-human clinical trial to test those antibodies. And since we were able to license those antibodies, so they have a good potential to be able to reach patient care and be used as a prophylactic or therapeutic medicine.
A central pillar of modern medicine is the ability to monitor the effectiveness of our interventions. And yet in cancer, there are large swaths of time throughout the patient's clinical course when we're operating in the blind. Recently, it has been discovered that tumors shed small part of their DNA into the peripheral blood. That circulating tumor DNA offers the possibility of monitoring tumors non-invasively through a simple blood draw. The challenge is that this is a very sparse signal. We have rethought this problem, and using advanced machine learning, we scan the entire genome for the tens of thousands of mutations that are found in human cancer. This allows us to track responses in real time in patients treated for metastatic melanoma with immunotherapy, and also to identify patients treated with surgery for lung cancer or colon cancer, and identify those individuals that have potentially lethal residual disease that was left behind after surgery to stratify this individual for additional chemotherapy or immunotherapy.
Environmental enteric dysfunction, EED, is a disease of the small intestine that is thought to be a risk factor for growth stunting, a condition that affects over 150 million children. EED leads to disruption of the intestinal barrier and loss of absorptive surface area. However, since most children in low-income settings do not have access to endoscopy, and there are a lack of validated non-invasive biomarkers, the etiology and prevalence of this condition is largely unknown. The Bangladesh EED study, BEAD, was designed to identify biomarkers of this condition and assess the relationship between the small intestinal microbiota and stunting in 12 to 18-month-old undernourished children. In this study, Children whose stunting was refractory to a nutritional intervention were eligible for endoscopy with collection of duodenal biopsies and aspirates. Using culture-independent methods and aptamer-based proteomics, we found that the abundances of several duodenal bacteria from children with EED were positively correlated with biomarkers of intestinal inflammation and negatively correlated with linear growth. Finally, to assess causality, we colonized germ-free mice with a consortium of duodenal bacterial strains obtained from stunted children from the BEAD study. Compared to control mice, those that were colonized with the BEAD consortium developed an enteropathy similar to that of the children with biopsy-confirmed EED. Understanding the mechanisms by which members of the small intestinal microbiota affect mediators of linear growth will facilitate the development of new therapeutic approaches for stunting, including those that are designed to reconfigure the small intestinal microbiome towards a healthy state.
The Ischemia Trial was designed to study whether adding invasive procedures to medical therapy improved outcomes for stable patients with coronary artery disease and markedly abnormal stress tests. The invasive procedures were an angiogram to take pictures of the coronary arteries, inserting a stent to open narrowed arteries, or bypass surgery. We found no overall difference between the two groups in cardiac event rates, but patients with chest discomfort or angina at baseline, assigned to the invasive group, had more improvement in quality of life.
Our findings show that we do not need to rush patients with coronary artery disease to undergo procedures before a trial of good medical therapy. Our results do not support the common practice of doing these procedures in patients with mild or no symptoms. The trial highlights the importance of shared decision-making between physicians and patients and should result in less stenting and cardiac bypass surgery performed.
For most people, puberty is something that starts naturally on its own. But pediatric endocrinologists like myself often see children who haven't started puberty by the typical age. A challenge in evaluating these children is that we have no reliable method to predict which children will eventually go through puberty on their own, which most children will, and which children have a rare pathological problem with the reproductive endocrine system. To address this problem, we've developed a new diagnostic test called the Kisspeptin stimulation test. In our study, we administered this test to 16 girls and boys with delayed or stalled puberty to determine who would respond to Kisspeptin and who wouldn't. We then followed these children until age 18, following some for up to 4 years, to see who would eventually enter and progress through puberty. We found that 8 of these 16 children had clear responses to Kisspeptin, and these 8 children eventually progressed through puberty. The other 8 children did not respond to Kisspeptin robustly, and none of them entered puberty on their own by age 18. So, in this cohort of children, the Kisspeptin stimulation test accurately predicted which children would progress through puberty and which would not. We're now planning a larger study to determine the accuracy of the Kisspeptin stimulation test, and we anticipate that the test will be an invaluable clinical tool for evaluating and managing children with delayed puberty.
The ongoing pandemic of coronavirus 2019, also known as COVID-19, represents an unprecedented public health challenge. Given the initial reports of high morbidity and mortality early in the pandemic, we began developing a treatment study for this infection even before there were any cases of COVID in the United States. The Adaptive COVID Treatment Trial, or ACTT, was the first large randomized placebo-controlled trial of an antiviral for the treatment of COVID-19. The drug we tested is called remdesivir, which is a medication that affects the virus's ability to replicate inside the cells. In our trial, we demonstrated that remdesivir was effective at treating COVID-19. The main outcome we measured was the time to recovery, which we defined as the amount of time until someone could be discharged from the hospital. Those that received remdesivir had a time to recovery of 10 days as compared to 15 days in those that received placebo. The largest benefit was seen in those on oxygen, but there was benefit in the other groups in the study as well. In the middle of a pandemic, we were able to start the study in just a few weeks. The study was conducted at 73 sites in 10 countries and ultimately enrolled 1,062 participants in just 59 days. The study has changed how we treat COVID-19. The data from this trial supported the emergency use authorization of remdesivir in May 2020 and the final FDA approval in October 2020. This was only possible given a motivated group of networks and fantastic collaborators that came together to address this unparalleled public health emergency.
Vitiligo is a disease where the immune system attacks the pigment cells or melanocytes in the body. This leads to white spots, often in exposed areas such as the face and hands. Patients experience decreased quality of life and social stigmatization. Despite affecting about 0.5 to 2% of the world's population, no medications are FDA approved to repigment vitiligo. We helped evaluate ruxolitinib cream to see if patients may be repigmented. About half the patients who received the highest dose of the medicine repigment 75% or more on the face. A significant number of patients also repigmented on the body as well. Patients saw a response as early as 8 weeks after starting treatment. As a topical, this medicine was well tolerated with a favorable safety profile. It is currently being tested in a phase 3 program to hopefully become the first medicine that will be FDA approved to treat vitiligo.
Neurofibromatosis type 1 predisposes to tumors called plexiform neurofibromas, which can grow along any nerve in the body. They frequently occur in very young children, grow relentlessly, and cause problems including pain, disfigurement, and vision loss.
In the absence of effective medical treatments, we conducted several clinical trials with targeted agents, which were unsuccessful until we identified that the MEK inhibitor, selumetinib, shrank the majority of plexiform neurofibromas in children and improved pain, disfigurement, and function. Selumetinib became the first FDA-approved drug for NF1, which is a great advance.
Hi, my name is Philip Moss. I've been on the selumetinib trial for almost 6 years now. The way selumetinib has impacted my life is greatly. My tumor used to affect my physical appearance on my neck very greatly. And now people don't even notice it. So I used to get questions all the time about what's on my neck or am I doing alright? And now I just fit in like a normal kid.
We are grateful to the NF1 community, our participating sites, and patients and families who participate in NF1 clinical trials.
The CENTAR trial is a milestone in our fight against ALS. The project was a beautiful collaboration between a pharmaceutical company, Amylyx Pharmaceuticals, and the ALS academic community. The project was funded in part by the Ice Bucket Challenge. ALS is also known as Lou Gehrig's disease. It's a neurodegenerative disease, and people with ALS have progressive muscle weakness. This means that they lose the ability to do the things that we do every day that we take for granted, like walking and talking. In CENTAR, we tested a new investigational product called AMX35. AMX35 protects against nerve degeneration by using a combination of two medications that each target a different cell component, the mitochondria and the endoplasmic reticulum, respectively. The excitement is that people who took the study drug retained physical functions longer, like speaking, swallowing, walking, and they survived several months longer. This is the first drug we have tested that has positive effects on both function and survival in ALS. This is very hopeful news for people affected by ALS. We look forward to continuing this line of research and hope that this is just the beginning of new treatments for ALS.